What is the history of RSV vaccine development? Imagine a world where a tiny virus causes big trouble for little lungs. That's what respiratory syncytial virus does, especially in young children. The journey to develop a vaccine against RSV started back in the early 1960s, shortly after scientists identified the virus. The first big attempt was a vaccine made from inactivated virus treated with formalin, called a formalin-inactivated RSV vaccine. It was tested in infants and young children, but instead of protecting them, it caused a serious problem called vaccine-associated enhanced respiratory disease. When children who had received this vaccine later caught natural RSV, many developed worse symptoms like wheezing, fever, and lung inflammation. About 80% of these children needed hospitalization, and sadly, 2 children died from severe lung issues. This setback made researchers very cautious and slowed down vaccine efforts for many years as they tried to understand what went wrong. Scientists discovered that the formalin-inactivated vaccine caused an abnormal immune response. It produced non-protective antibodies and triggered a Th2-skewed immune response, which led to harmful lung inflammation when children encountered the real virus. This understanding helped guide safer vaccine designs later on. In the 1990s, efforts shifted toward passive immunization, which means giving protective antibodies directly instead of a vaccine. Titer RSV immunoglobulin and monoclonal antibodies like palivizumab were developed to protect high-risk infants, such as those born prematurely or with weakened immune systems. Palivizumab has proven to be safe and effective, but it is expensive and needs monthly injections, so it is mainly used for the most vulnerable babies. In recent years, advances in molecular biology and structural virology have really sped up vaccine development. In 2013, scientists uncovered the detailed structure of the RSV fusion protein, a key part of the virus that helps it enter cells. They found that stabilizing the perfusion form of this protein made it much more effective at triggering an immune response. This breakthrough led to the creation of new vaccine candidates that are more likely to protect children from severe disease. Today, researchers are exploring various types of vaccines, including protein subunit vaccines, live-attenuated vaccines, vector-based vaccines, and even mRNA vaccines. Over 30 of these candidates are now in clinical trials, showing how far the field has come after decades of challenges. In 2023, the U.S. Food and Drug Administration approved RSV, the first RSV vaccine licensed for use in adults aged 60 and older. This milestone is a big step forward because it helps protect older adults from serious lung infections caused by RSV. While this vaccine is for adults, it also opens the door for developing safe and effective vaccines for infants and young children, who are most at risk of severe illness. The history of RSV vaccine development spans more than 60 years, starting with a failed vaccine that caused harm, then moving through many years of research to understand immune responses better, and now reaching a point where modern science offers hope for safe, effective vaccines. Always remember, if you or your child are considering vaccination, it's best to talk with a healthcare professional to get advice tailored to your individual needs.